Okay, for smooth labor. Okay, and also due to any accidents. So if there is any harm for the baby, so it will prevent from the injury mostly. And it also prevents from lung collapse, and also it opens digestive tract. Then the next one is allantois. So allantois originates from gut. Okay, and it forms the umbilicus, that is umbilical cord, which will have blood vessels, and it is also a reservoir for the nutrients and the waste which are released by this fetus. So there is another membrane which is called chorioallantois. So this is due to the fusion of two membranes, that is allantois and chorionic membranes. So there is chorionic membrane, that is the outermost membrane, which is directly in contact with the uterine tissue. Okay, it has the allantoic vessels, and it is the site for hormone production, nutrient and gas exchange. So during pregnancy, there are lots of there is important role of hormones from like growth as well as delivery, then milk production. So everything, contractions, uterine contractions, so everything is controlled and regulated by these hormones. Okay, so everything, the production of these hormones is in the chorion membrane. So there are total 4 fetal membranes which will develop into conceptus. So conceptus is nothing but the term is used when we discuss about fetus along with the membranes. So there are 4 fetal membranes. 2 arises from the trophoblast layer of blastocyst and 2 arises from the inner cell mass of the blastocyst. So first of all, what is blastocyst? So after fusion, like fertilization of egg and sperm, so it will form a zygote. So this zygote will be dividing continuously, and it will form a ball of cells which is called blastocyst. So this blastocyst has the outer membrane which is called trophoblast, and inner you have a mass of cells which are called inner cell mass. So from this outermost trophoblast, there are 2 membranes which are arised, which are continuous with the somatopleurae of the embryo, and from the inner cell mass, there are 2 splanchnopleural membranes which are in continuation with splanchnopleural layer of the embryo. So these are vascular in nature. So if you see this structure, so you have the outermost layer which is called somatopleurae, and below that you have chorion, and inside you have the splanchnopleural, which is surrounded by the yolk, which is surrounding yolk sac. And here in this area where the embryo is growing, you can see it is surrounded by amnion or amniotic fluid. Okay, and to that you have a membrane which is called, surrounding that, it is allantois. So what is this chorion? Chorion forms the outer boundary of the entire conceptus. So whatever the fetus which is present along with the membrane, so the outermost membrane is called as chorion. This is arises from the trophoblast layer. So it's the outermost membrane, and it is in direct attachment to the mother. Then next is amnion. So amnion, it encloses the embryo within fluid-filled amniotic cavity, and it is formed by folds of chorion. Okay, so in domestic mammals, usually if you see in humans, in domestic animals, you can see the folding of chorion will form this amniotic cavity. But in humans, you can see it is by the trophoblast. They will form a cavity. And it is non-vascular. You have a fluid here which is called amniotic fluid. And this fluid is produced by fetus. And it is a protective cushion for the baby or fetus. And during birth, so usually that is called a breaking of water. That means whatever this amniotic fluid is there, that will be coming out. That membrane will be ruptured, and that fluid comes out. The next is allantois. So allantois develops as an outgrowth of hindgut. So hindgut is a backside. Okay. So hindgut splanchnoplurae, which originates from the inner cell mass, and it grows to fill an entire extraembryonic coelom with a fluid-filled allantoid cavity in the domestic mammals. So the outermost surface of the allantois binds with the inner surface of the chorion. Okay, and it is highly vascular and it has the vessels. Okay, vessels of the placenta. So from the placenta, whatever the blood vessels comes, so that will be present here, and it fuses with the chorion sometimes, which is called as allantochorion or chorioallantois. This brings blood vessels to the chorion. Next is yolk sac. So yolk sac is continuous with the midgut. Sorry. So it is continuous with the midgut, and that midgut is splanchnoplurae, and it develops early in hypoblast formation from inner cell mass, and it is supplied by vitelline vessels, and it forms early temporary placenta. Okay, so in horse and dog, if you see, it will form a temporary placenta, and this yolk sac is most important in egg-laying vertebrates. So if you see, it is not much, yolk is not much important in the placental mammals because there is a placenta which will be supplying the nutrients from mother to fetus. But in egg-laying vertebrates, so as they grow outside the mother, okay, so this yolk provides all the nutrients from hatching till birth. Okay, and as I said, conceptus means embryo or fetus along with the fetal membranes. And in birds, this yolk sac will nourish the embryo. And in mammals, they will provide blood cells and also primordial germ cells. So what are these primordial germ cells? They are nothing but the germ cells which are going to become eggs or sperm in the future, okay, of that organism. So this is the structure, if you see. So the outermost membrane, you can see chorion, which is somatoplurae, and inside you have amnion. Excuse me. Amnion. Within the amnion, you have the amniotic cavity, and that there the fetus is growing. So you have hindgut and heart there, and embryonic coelom is there at the end. And towards this side, you have the vitelline membrane which surrounds yolk sac, and allantois which surrounds the allantoic cavity. Now, what are the developmental adaptations of amniotes? So, where the amniotic cavity is present, those animals are called as amniotes. And the embryos of birds, reptiles, and mammals, they develop into fluid-filled sac in the uterus. And such organisms are called as amniotes. They have some adaptations. So, here there are 4 extraembryonic membranes which form around the embryo. So, as we have discussed, chorion, amnion, yolk sac, and allantois. So this chorion is the main function is to exchange the gases. And amnion, it encloses the amniotic fluid. Then yolk sac encloses the yolk. And allantois, it disposes the waste product and contributes to gas exchange. So even the fetus will be producing the waste material throughout its development. So that is collected and disposed by this allantois. So if you see here in birds, you can see the outermost, if you see the egg, chicken egg, okay? So you can see outer, you have a shell. And after that, you have the albumin layer, okay, which is made up of a protein. Then inside that, we have allantois, Okay, so chorion and allantois structure. So allantois is surrounded by the chorion. Then you have amnion, and you have amniotic cavity with an amniotic fluid, and in that you have the embryo. And you have yolk sac, and within that you have yolk. So that yolk provides the nutrients. So this is the unincubated chicken egg. You can see it is surrounded by shell and outer shell membrane. Then you have intershell membrane. So within that we have watery albumin. And that albumin is also, if it towards the center, it is viscous in nature. And we have some membrane called chalaza. Then at the center, you have cytoplasmic island, and inside that you have yolk. So this cytoplasmic island is surrounded by vitelline membrane. So the structure of egg is, again, it is 4 membranes. Chorion, which is the outermost membrane, which aids in the gas exchange. Then allantois exchanges oxygen and carbon dioxide and stores the waste material. Then amnion. So it's the amniotic sac containing the amniotic fluid, which surrounds the embryo. So this will be a shock absorber as well as cushion for the embryo. Then the yolk sac. So it is the source of food, yolk. It's rich in nutrients, and that will provide blood vessels which transports the food to the developing embryo. Then this is how the chicken eggs look like. So the outer thin layer of albumin, okay, which is present at the center. And inside that we have a germinal disc. And towards on 2 sides of this albumin layer, we have yolk and chalaza. Then we have outermost shell, then inner shell membrane, outer shell membrane, then yolk membranes and albumin, and we have chalaziferous layer. So all this we have discussed now. So like human beings has 10 months of gestation. So even the incubation period is different for different animals. So in the chicken egg, so we can see it is 21 days of incubation and it is hatched on 21st day. Okay. So after 24 hours of fertilization, neurulation will start. So neurulation is nothing but the formation of nervous system. So this is how after 24 hours it looks like. So here you can see developing brain, then neural folds, neural groove, and you have spinal cord, small button-like structures of neural tube. So from neural tube, nervous system will develop. And we have primitive streak, which usually seen in birds. Then after 48 hours, we see the structure like this. We have a developing eye, developing brain, then atrium, ventricle that forms heart, then developing ear, and also spinal cord, somites, which will form germ cells, and vitelline blood vessels. Then after 56 hours, it looks like this. Then after 96 hours of incubation, so we have a heart now, ear, eye, brain, anterior limbed, posterior limbed, allantois, and a tail. So it forms a nice structure which is developing. So the same thing which we have discussed of the fetal membranes in the thing. So this is how it looks like. We're not going in depth of development, but what are the fetal membranes which are present and what are the functions of those fetal membranes in chick. Now we see the functions of placenta. The placenta usually seen in mammals, placental mammals, and what are the classification, structure and functions. So placenta is a temporary organ which is seen only during pregnancy. So it is connected to developing fetus via umbilical cord. So its main functions are to supply nutrients, thermoregulation, waste elimination, gas exchange, and from the mother's blood supply, and to fight against internal infections and produce hormones which supports the pregnancy. And the placentas are a defining characteristics of placental mammals. So only in placental mammals we see this placenta, but rarely we also see in the primitive animals like marsupials and some non-mammals also. But it is not the prime thing there. And the placentas of all the eutherian mammals provide common structural and functional features. So they have some common structure and functional features, but there are some differences also. So there are two characteristics are particularly divergent and form basis for the classification. So if they are all uniform, there is no need for classification. If we are classifying means there are some differences which are present in different organisms. So what are the differences are mainly in the shape of the placenta and the distribution of contact sites between fetal membranes and endometrium. So endometrium comes from the mother and fetal membranes. So this what kind of interactions are there between fetal membrane and endometrium, and number of layers of the tissue between mother and fetal vascular system. So these are the main things on basis of which we are classifying the placenta. So first is on the basis of mode of implantation. So what is implantation means? So usually in females, so they'll have a lining which is around the uterus, which is called as endometrium. So this lining will be usually removed every month as a part of menses. Okay, whenever it is removed, so then it results in the menses, menstrual cycle. So whenever a woman gets pregnant, so the fertilized egg will go and implant in this endometrium. So endometrium, it provides the support and also nourishment. So where till the placenta is formed, so the nourishment is provided by endometrium, and also it provides the support so that there the embryo will grow into the fetus and then finally the complete organism. So here, based on the type of implantation, this placenta is of three types: superficial, eccentric, and interstitial. So superficial we see in dog, monkeys, and carnivores; eccentric in squirrels and rats; interstitial in hedgehog, ape, humans, bats, guinea pigs, etc. So we will look into the details. So in the superficial implantation, the growth of the chorionic sac brings it into contact with the lining of the main uterine cavity. And this type of implantation is called central implantation. That means we have this embryo which has a chorionic sac. So this chorionic sac is in direct contact with the uterine cavity. So here if you see the structure. So this chorion, the outermost membrane is chorion. That chorion is in direct contact with the uterine cavity. So then that kind of thing is called a superficial implantation. And then is eccentric implantation, where the chorionic sac lies for a time in the fold or a pocket, which loses off from the main cavity. So here, the yolk sac is in the pocket. Okay, it is inside. And the uterine cavity, there is a separation. Okay, then interstitial implantation is the chorionic sac penetrates into a substance of the uterine lining. So here you have a lining. Embryo is at the center and it's a wall of uterus, and in between you have a lining. So this kind of implantation is called interstitial implantation. And on the basis of origin of villi, villi are nothing but a finger-like projections. So that we'll discuss in detail. So based on that, we can divide the placenta into, you have yolk sac and elentochorionic type of placenta. And in the elentochorionic, in yolk sac, you don't find all these things. Then in Allentochorionic placenta, we have again non-deciduous and deciduous. And the subtype is called Contra deciduate. So in the non-deciduous, we have diffuse, cotyledonary, and intermediate. And in deciduous, we have zonary, discoidal, and metadiscoidal. So diffuse type of placenta is seen in horses and pigs. Cotyledonary is seen in ruminants. Ruminants are nothing but cows, sheep, deer, all this. Then intermediate are seen in giraffe. And in the zonary, you have dogs and cats, discoidal, rodents and primates, and metadiscoidal humans. So here we can see various membranes. You can see here vitelline blood vessels are seen here, but here you can see in the yolk sac, you can see the vitelline blood vessels, whereas in the allentochorionic, we see allentonic blood vessels. Allantois is present here. So first is non-deciduous placenta or semi-placenta. So in most mammals, the implantation is superficial. That is, the blastocyst, that small ball of cells, lies in the cavity of the uterus in contact with the uterine wall. And the contact may be made more intimate by the surface of the blastocyst by forming a finger-like growths, which are formed from the trophoblast, and they are called as chorionic villi. So that means from the blastocyst, small finger-like projections comes. The trophoblast is the outermost layer. So from trophoblast, small finger-like projections comes, and they are called as chorionic villi, which will be penetrating into the uterine wall. And the blood vessels of chorionic villi are the branches of allantoic blood vessels, if you see in chorioallantoic placenta. During the birth, what happens? Along with the baby, when placenta will be delivered. And this chorionic villi are simply drawn out of the depression. Okay, where they have inserted into the uterine wall, they simply come out. There is no damage to the uterine wall, and there is no bleeding also occurs after the delivery. And this type of placenta is called non-deciduate or non-deciduous placenta and is seen mostly in pigs, cattle, and some other mammals. Further, the chorionic villi is a non-deciduous placenta. It lies in apposition with the endometrium. So it doesn't fuse with it. So then in such case, it is called a semi-placenta. The next is deciduous placenta or placenta vera. So it is the degree of intimacy between the maternal and fetal tissues is increased. That means they are more strongly associated. And the wall of the uterus become more eroded with the action of the trophoblast, and they penetrate into the uterine wall very strongly. Therefore, when this chorionic villi fuses with the eroded uterine mucosa, that's why it is called true placenta. And whenever there is delivery or parturition, so whenever this placenta comes out, there will be more or less hemorrhage from the uterine wall. That is, bleeding can happen. And such type of placenta are found in higher uterine mammals, which are called deciduate or deciduous placenta. And the maternal tissues are expelled at birth. In case of decidual placenta are called as decidua. And the hemorrhage will be stopped by the same mechanism where the expulsion of newborn takes place. That is contractions of the muscular wall, which will slows down the blood flow and until the clotting of blood stops the hemorrhage. Okay, so therefore it will try to prevent the bleeding or blood flow. Then contra deciduate placenta. So this is seen in mole. Okay, it is a modified type of deciduate placenta, which is called as contra deciduate placenta. So here, not only there is a loss of maternal tissue, but also the fetal portion of the placenta is also lost. Okay. So in the non-decidual placenta or semi-placenta, we have again different types. One is diffuse placenta. So this is seen in some mammals like pig, horse, mares. So where the chorionic villi remains scattered all over the surface of the chorion. Okay. And then is cotyledonary placenta, where they are found as a patches. Okay, then you can see that chorion surface is not smooth. There'll be like rosettes, like you can see small patches all over the chorion. And this is usually seen in ruminants, like cattle, sheep, deer. In that animals, we see this type of placenta. The next is intermediate placenta, which is rare type, and it shows free villi or cotyledons. And they are arranged, like they are scattered all over the chorion, and this is, hence it is called intermediate type of placenta. So in these three types of placenta, during parturition, the fetus will not damage the uterus. Okay, therefore it's called non-deciduous placenta. This is seen in camel and giraffe. Then deciduous placenta or placenta where other is true placenta, where first is zonary placenta. So here the villi is developed in the form of a belt or griddle-like band around the middle of the blastocyst or chorionic sac, which is more like elliptical in shape. The placenta, this type of placenta occurs in carnivores like cats, dogs, and raccoons have this incomplete zonary placenta. Then another one is discoidal placenta, where it is seen in insectivores like bats, rodents, like mouse, rat, rabbit, and they form circular disc or plate on the dorsal surface of the blastocyst. Then third one is metadiscoidal placenta. So in primates, this discoidal placenta is found, but they are special type. They are first scattered, but later becomes restricted to one or two discs. So if you have monodiscoidal, then only single disc is seen. Disc, okay? This is seen in man usually. And bidiscoidal means in monkeys, we see two disc-shaped areas. Such as, like in monkeys, we see such kind of placenta. Then based on the histology is nothing but the study of tissues. So based on the tissues, this placenta is again classified. So usually there are 6 tissues which will be participated, or 6 membranes which are participated in the placenta formation. So they are the endothelium of the maternal blood vessels, endometrial connective tissue which is mesenchyme, and uterine epithelium. These 3 are from the mother side, and the ectoderm of the chorion or chorionic epithelium, then chorionic connective tissue or fetal mesenchyma, and the endothelium of the fetal blood vessels. So these 3 are from the fetal side. So 3 membranes are from the maternal side and 3 membranes are from the fetal side. So here they are classified into 5 types: Epitheliocorial placenta, Syndesmo Corial placenta, Endotheliocorial placenta, Hemophorial placenta, and Hemo-endothelial placenta. So there are 3 layers of fetal extraembryonic membranes in the chorioallantoic placenta. So they consist of, as we have already discussed, endothelium lining of allantoic capillaries, connective tissue in the form of chorioallantoic membrane, mesoderm, and the chorionic epithelium, the outermost layer of the fetal membranes derived from the trophoblast. And the other 3 are from the maternal side, but the number of these layers which are retained but not destroyed in the process of placentation. So these 3 layers are endothelium lining of endometrial blood vessels, connective tissue of the endometrium, and endometrial epithelial cells. So in this, we will see how many membranes are participating. So in the epitheliocorial, we have 3 layers or 3 membranes from the fetal side and 3 membranes from the uterus. So these are usually seen in marsupials, ungulates like pigs, horse, cow, cattle. Okay. Then Syndesmo Corial, where 3 layers from the fetus and 2 layers from the uterus of the mother participate. And examples are cattle, sheep, ruminants. Then Endotheliocorial. So where 3 layers from the fetus and 1 layer from the uterus participates. So these are seen in carnivores like dogs, cats, bears. Then Hemophorial placenta, where 3 layers from the fetus and no layer from the, or no membrane participates from the uterus side. They are primates or insectivores. And Hemoendothelial type of placenta, where fetal layers are two fetal layers are participating and no membrane from the uterus side are participating. So in the higher rodents like rat, guinea pig, rabbits, so those are the examples. So this is how they look like. So first is the endothelium, mesoderm and trophoblast, and below is epithelium, connective tissue and the endothelium of the mother. Above one is from fetus. So in the epitheliocorial, all the 6 membranes are participating. In epitheliocorial, you can see only 5. The middle ones are fused. Then in the endotheliocorial, you can see only 4. In the hemophorial, you can see only 3. Okay, this is how it looks. So this is the original one, like other description, other type of description. Now we will discuss each one. Epitheliocorial placenta. So this is the primitive type and it's found in marsupials, pigs, horse, cows, etc. So here all the 6 tissues are participating. 3 from the mother and 3 from the fetus. And the immediate contact of the 2 halves of the placenta involves chorionic epithelium and uterine epithelium. They will be forming a like border. So that's why it's called epitheliocorial placenta. And the villi of the epitheliocorial placenta push into the wall of uterus and they'll form in a pocket-like depressions in the uterine wall. So here we can see. So the fetocorion is in contact with epithelium of the uterus, hence it is called epitheliocorial placenta. So here fetal and maternal 6 layers are present. Okay. Then Syndesmo placenta, which is seen in ruminants, and here the fetal and maternal components are fused. Therefore, the uterine epithelium is destructed completely. So the chorion is in direct contact with the connective tissue of the uterine mucosa. Here we can see only 5 tissues which lie between the 2 blood streams. Okay, this is called Syndesmo placenta. And this chorionic villi will pierce into the uterus of the mother and the chorion will come in contact with the syndesmo of mother's uterus. Hence it is called syndesmos corial. The next is endotheliocorial placenta, which is seen in carnivores. So here the uterus mucosa is reduced and chorionic epithelium comes in contact with the endothelial wall of the mother uterus. Only 4 barriers are seen from the mother and fetus, and this type of placenta is called endotheliocorial placenta. And the chorion of this fetus will come in contact with the endothelium of the mother's uterus. Hence, it's called endotheliocorial placenta. Then fourth one is hemophorial placenta, seen in primates, insectivores, and bats. Here only 3 layers are seen. The endothelial walls of maternal blood vessels also disappear here. And the chorionic epithelium is directly birthed in the maternal blood sinuses. Okay. So endothelial lining is completely absent. And the placental connections are more intimate. And the fetus will float in the blood pool of the mother's uterus. Hence it is called as hemochorial. Then last one is hemoendothelial placenta. So in the hemoendothelial placenta of higher rodents, the number of barriers are still reduced. Now only you have 2 types of membranes which are participating and they lose their epithelial connective tissues, connective layers from both the sides. And the bare endothelial lining of their blood vessels alone separates the fetal blood and the maternal blood. So these are the various membranes which will be participating in egg-laying animals and placental mammals. So during placental formation, so what are the membranes which are participating and what are the classification of placenta? And in the egg-laying animals, so what are the layers which are present in the egg which are protecting the organism? So these are regarding the fetal membranes. So we'll continue with the next topic in the next class. Thank you.